Hello, and welcome to the fourth User Day. Please welcome me in welcome Dr. Paolo Ronchi and Dr. José María Andrés. Dr. Paolo Ronchi, Translational Chemistry and Technology Innovation Unit Lead at Chiesi Farmaceutici, if I pronounce that correctly. He earned his PhD in chemistry from the University of Milan in 2007, working on carbohydrate analogues, total synthesis. Paolo completed postdoctoral at the Università di Milano first, and then at Politecnico di Milano after. In 2011, Paolo joined the Boehringer Ingelheim Research Italy Institute as a medicinal chemistry research associate, and since 2013, he's been with Chiesi Farmaceutici, applying his expertise in organic chemistry. In 2022, he became the head of the Translational Chemistry and Technology Unit, focusing on the chemical aspects of drug discovery. I also would like to introduce José María Andrés Castán. He is a customer consultant for life sciences at Elsevier. He's responsible for supporting R&D scientists and engineers in their use of reactions across Europe, and joined Elsevier in 2023. Previously, José spent 10 years as a researcher in academic and private institutions in France and Spain, working on nanomaterials, material sciences, and organic chemistry. José holds a degree in organic chemistry and a bachelor's degree in chemistry. With this, I'll hand over the floor to Dr. Ronchi for his presentation, Unnatural Amino Acids, Overcoming Synthesis Challenges for Therapeutic Advancements. Over to you.
Thank you, Eric. First of all, I would like to thank you, Elsevier, for giving me the opportunity to present at this very interesting virtual congress. So I am Paolo Ronchi, and I lead the Translational Chemistry and the Innovation Technology Unit at the Chiesi Group in the Medicinal Chemistry Department. Before to go to the scientific part, I would like to point out the difference between creativity and innovation. Creativity is the ability to create new and potentially valuable ideas, when innovation is when these ideas are integrated into an already existing process, generating a competitive advantage. As we all know, the drug discovery is a very time-consuming and expensive process, and every day new and more strictly conditions are added to this process, and this forces pharmaceutical industry to innovate it in order to let it be still efficient and sustainable. It is a multidisciplinary process where chemistry, more precisely organic chemistry, plays a fundamental role at two different stages. In the early phase, supporting medicinal chemistry for the exploration of the molecular space and the identification of the candidate drug, while in a later phase, supporting the process chemistry to the identification of the best way to production the clinical candidate to feed the clinical phase and up to the eventual market landing. So pharmaceutical industry is always looking for new technology, reactivity, and approaches. And here I have collected two papers. The first one claims the central role of organic chemistry in drug discovery process, while the second is claiming where these innovative approaches and the idea can come from, and in particular from the interaction between academic world and the industry. But I want to focus on the impact that can have organic chemistry in medicinal chemistry. It can have a big impact on two different sides. The first one is quality. For example, giving the access to an unprecedented molecular space that can lead to the identification of a better clinical candidate with a lower off-target effect and with a better Physchem and ADME profile. Last but not the least, having an IP-free status. The other side is quantity. The speed of synthesis of compound is a very important parameter, and if you can synthesize more molecules in a given time, you can better feed up the medicinal chemistry project. And this will lead to a speed up clinical candidate identification. These two factors will lead together to a higher sustainability process. But what are these new ideas, technologies, and approaches that pharmaceutical industry are looking for? I try to summarize these into these slides, dividing them into three different classes: activity, technology, and innovative approach. But based on my experience, only when you are combining elements coming from these three different classes of ideas, you can have a disruptive innovation on the medicinal chemistry project. In the next slide, I will show you a study case where some of these elements had been combined to generate innovation. As you all know, amino acids are characterized by two different functional groups, and they can be easily functionalized. Moreover, they have a stereogenic center, and these reflect their high sp3 character, and they are abundant in nature. All these features make them very useful for drug discovery project, especially when you are targeting the 3D based molecular space. Basically, when you are escaping from the flatland. So I have listed the 20 amino acids, dividing them regarding their nature of the substituent. Those having sp3 substituents and those having sp2 substituents. They are 20, and you can design and synthesize a huge amount of peptides. But if you want to carry out a peptide optimization, maybe you need to have access to a new molecular space. And this can be done using non-canonical amino acids that are still amino acids, but they are not present in nature. In the next slide, I have tried to summarize different type of non-canonical amino acids. The first one is when you are using amino acids having an opposite configuration of the stereogenic center. For example, using D-series amino acids. The second one is when you quaternarize the stereogenic center, like by alkylation. This is very important amino acids, but they are rarely commercially available because the difficulty of their synthesis, and moreover, you have to be very good at the controlling at the newly quaternarized stereogenic center. The third one class is the homologation, when you put some methylene group between the amino and the carboxylic acid. This is easier to synthesize, but you have still to be able to control the stereogenic center. The third one is the unnatural amino acids, and is when you are using amino acids with a different substituent group that is not present in nature. And this is the case that I'm going to present you. In our medicinal chemistry project, we had the need to synthesize analog of phenyl, alanine, tryptophan, tyrosine, and histidine in order to generate a small library of sp2-based unnatural amino acids. And based on our disconnection, we thought that the key bond to disconnect was this sp2-sp3 bond. To tackle this bond, you can follow two different strategies: the dipolar approach or the radical approach. So we went to the literature looking for any paper that pursued this one of these two approaches. And we use a Reaxys, that is a powerful tool to carry out the research. And we found basically using the polar approach, the bond could be disconnected by sp2-sp3 coupling, for example, like a Negishi coupling, or the total synthesis of the entire amino acids. I have collected an example coming from a patent where they synthesized the thiodiazole unnatural amino acid. But as you can see, when you are using the polar approach, it could require many steps you have to use many functional group manipulation, and you have to be very careful about the reaction condition, because you have to be functional group tolerability, and also you have to use a protecting group, and this led a very low sustainable process. Moreover, you have to be able to control the newly formed stereogenic center if is forming during the reaction. So we went back to literature using Reaxys again to find if any other author used a radical approach to do this unnatural amino acid. And we found that a patent that they used to synthesize unnatural amino acid via nickel radical cross coupling using an N-hydroxyphthalimate derivative of various glutamic and aspartic acid in presence of the zinc dust, that is the reductive agent that can generate the radical. So they managed to synthesize a big collection, 200 amino acids, with one single step for each amino acid, leading a very high sustainable process. But if they were synthesized a huge amount of amino acids, these amino acids were poorly functionalized. They used only a relay substituent, very low functionalized, and a nitrogen base 6-member ring. But we had the need to use a broader range of heterocycle, also 5-member and also electron-rich heterocycle. So we came across to this very interesting and important paper of Professor Baran, where they carry out this nickel radical cross coupling using electrochemistry, in particular using an electrode of magnesium that is the anode, and it is a sacrificial electrode, and as a counter electrode, the RBC electrode. So, inspired by one of the example reported in the paper, we build up our reaction model using as a radical precursor this aspartic acid derivative, always an hydroxyphthalimide derivatives, and as electrophiles this para-iodo benzaldehyde, because aldehyde is a very sensitive functional group, and you can test directly your tolerability, your functional group tolerability, and moreover, you can use it for further functionalization. Here I have reported the test condition that we tried in order to optimize the reaction. First, we replaced the nickel chloride with the nickel bromide, because nickel chloride is a carcinogenic agent and we were not allowed to use. And also we replaced the aryl iodide with the aryl bromide, because they are more commercially available and they are more atom efficient. So we try to avoid the use of nickel bromide and also silver nitrate that was used as additive in the original procedure of Professor Baran, but without any of these salts, no reaction was observed. So we also try to replace silver nitrate with iron acetate, and when we did this replacement, we obtained 20% of yield of reaction. Then we tried to replace the sacrificial anode magnesium with a non-sacrificial anode like a glassy carbon, but we didn't observe the formation of the desired product. But when we substitute the magnesium with the aluminium, that is still a sacrificial electrode but is cheaper, we obtain 40% of yield. So we did this exercise of replacement with the counter electrode, RBC, and trying different platinum-based electrode and graphite, we never observed the formation of the desired product. But when we use nickel foam electrode, we observe 20% of yield. At the end, we use also the combination nickel foam and aluminium as electrode, and we observed the 39%. That, if it is compared with the 42% of yield of the original procedure, we can say that aluminium nickel foam combination is a valid alternative if compared with the magnesium RVC electrode. So we decided to test in a head-to-head comparison of these two setups of the electrode with a broader range of heterocycle. And we applied this head-to-head comparison to this heterocycle. As you can see, we used both 6-member nitrogen-based heterocycle, both 5-member and electron-rich heterocycle. As you can see, for these 3 amino acids, we obtain higher yield using aluminium nickel foam electrode if comparing with those obtained with magnesium RVC. And we can say that aluminium nickel foam is a competitive setup of electrode because and they are also cheaper. I want to point out also that this last 3 example was obtained with a low yield, 10%, but this is a big difference to having even in low yield to not having them, because having it give you the opportunity to use them on a medicinal chemistry project, and you can explore the relative molecular space. We also check the optical purity of these amino acids, and we observed that we didn't lost any optical purity of the stereogenic center. So we decided to build up our scope using magnesium RVC combination because we decided that were more reliable than the newly found couple nickel foam aluminium. And we build our scope of 6-member and nitrogen-based heterocycle. As you can see, we were able to install different functional protective group on the sp3 part, and also we were able to install cyano group or methoxide group, fluorine atoms, and 6 nitrogen-based heterocycle, both we starting with from aspartic acid derivative and glutamic acid derivative. In this slide, I collected while the 5-member ring, and these are the first example of nickel radical cross coupling applied to aryl bromide of 5-member electron-rich heterocycle. As you can see, we managed to install different thiophene derivative, thiazole, having both methyl group or electron withdrawing group. We also managed to install pyrazole, thiodiazole, and oxadiazole using aspartic acid and derivative, and also thiazole and oxatiazole using glutamic acid derivatives. Some of substrates were not successful, like this isothiazole and these 2 triazoles. Moreover, we have scaled up this unnatural amino acid to 1.2 gram of product with a yield of 49% using ICA carousel that basically is 6 vials in parallel. Then we also try to functionalize this peptide having the free carboxylic acid in the lateral chain. We managed to synthesize the N-hydroxyphthalimide derivatives, and using our methodology, we were able to install this thiazole heterocycle. Here we obtain these beautiful crystals, and this led us to the confirmation that the optical purity was not eroded during these methodologies, and moreover, we open up the post-synthesis functionalization of small peptides, and this is the open access paper that we publish, where is contained all the work that I have just presented. And as last things, I would like to say thank you to my co-authors and you all for your kind attention. And now I leave the stage to Jose Maria.
Thank you, Paolo, for your great presentation. I would like to now change a bit gears, also piggybacking off on what you're saying. The idea now is, while I share my screen, the idea would be in your great work, you are working with electrochemistry reactions, okay? So what I planned for today is how if it is the other way around. If we have already specific conditions, as you had in your talk, what happens if we want to target that with a database, because we are that specific. So for that, I have prepared one specific example to go through it in Reaxys. It would be just this simple arylation of this pyrazine that is protected with well, several protecting groups, could be any of them. Okay, so the idea would be, how can I get to these specific reactions, but being in a procedure that's based on electrochemistry. So for that, I will need to jump to Reaxys first and do it from here. So I will just draw the input that I have already. That's, of course, how we would start. So I have my arrow for the reaction, and I need to specify my product. That is, of course, my pyrazine. And if you remember, it was just an arylation here. Now I need to design my protecting groups as well. That will give me the option of showing you a couple of things. For that, I have my R group option that will allow me to introduce a list of substituents over that position. Okay, as in this case, we had several protecting groups that we are considering for that purpose. We have the abbreviated groups placed here on top, so I could say, give me, in this case, this first one, then I can do both. Okay, I can do my list. I can be as exhaustive as I need to be. I don't need to add 4. I could add more if I wanted to. Okay, and then to complete the example, I would have 4. That's a representative case. Now, for Reaxys to understand that these 4 fragments are placed here, it's pretty simple. I just need to take my R tool again and drag and select them all. So now when I put my mouse, it understands that what I'm doing is just adding those 4 fragments, any of them at that position. I could also draw them or draw different things and use it in the same way. And I could specify also the attachment point with the tool that's placed just below the R. Now, for specifying the type of search that I do, I can claim, okay, I can add my starting material, or I can also use another tool that is called make break, in which I can specify which bonds need to be made in this case. So it will be this one. So this is the arylation, okay? And now if I can even go broader, I can even change the search strategy to go from a substructure. I don't really care if this benzyl ring is substituted. I just want to perform the arylation. Now I would be ready to go. I could transfer this reaction set to get to my results. As you can see, it's a lot of them, okay, 4,000. It's great in the sense that we have a lot of information, but now we need to understand how we can get to specific reaction conditions. Of course, apart from the 4,400 reactions, we'll have conditions with them, okay? So if I want to do that manually, it will be not that straightforward. We can do a lot of things already from the filters. That would be filtering by solvent, by yield, by anything that we can think of from here. But the condition need to be filtered out in the query builder, okay? So we need to perform the search again. Of course, we could randomly find the electrochemical reactions here, but I will show you a better way. The query builder, it's a white canvas in which we can filter off by reaction conditions. Could be like temperature, it could be anything that we can think of, okay? So what we can do is relaunch our search from scratch, and I will have the option of editing that into the query builder. as it is transparent and it will always show me what it did in the background, which in this case is pretty simple. It's just running that exact search. Now, I want to show you the filter where you can do this condition filtering. It's found here in the right. It's under topic and keywords, and it's the second field overall, which is reaction data and conditions. Here it will look into the information we have excerpted from the reaction and the experimental procedure if we have it as well in the database. Okay, so it's pretty powerful. So here I could say electrochemical. Okay, and it will find where that word is found combined with my reaction search that I did at the beginning to get to the 18 cases in which I have it. Okay, so it saves a lot of time when processing this information, and we will have it highlighted as we have it here. This is just one of the examples that you can use this for. Of course, you can use it for other types of reaction, green chemistry, or even dim stark, or it can be many of them, irradiation, microwave, and so on. I would like to show you a second example, which would be large scale, which can be also something that can be representative. In this case, it's quite straightforward. I can look for large scale if that's what I'm looking for, okay? And that would be already a good starting point. I'm getting 11 hits in which I have that, which is great already. However, I want to point out something about the query builder to end up. So here we have already our input, and it's great, but you can add multiple synonyms or different ways in which you would be happy with the results, meaning large scale is good, and that's a good way of defining a reaction that has been performed in a large scale. But also we may be happy with the reaction or the experimental procedure being performed in kilomoles or kilograms. So we can add synonyms in each line by adding a semicolon. So I could find kilogram or kilomole or industry scale, for instance. I could work on that and make it as specific as I want. The idea of making it more specific is that you can also save and export from this top icon, so you can even share it with other colleagues. But of course, the important thing of making the research more complete is that it will allow you to retrieve not only information on the experimental procedure based on one term, but you can add synonyms and make it more complete, such as in this case in which, for instance, we were missing. And it's already fine for us because we see that it's performed in a kilogram scale. I hope these tips on how to use the query builder are useful. And of course, do not hesitate to reach out to any of your Elsevier representatives if you have any question on this regard.
Thank you very much, Jose. And thank you very much, Paolo, for your presentation as well. We have questions in the chat. And let me start first with one that is, what are the main advantages that the new approach offers over traditional synthesis methods in creating drug-like unnatural amino acids for you, Paolo?
The main advantage is that you can create basically one unnatural amino acid in a single step of synthesis, even if it is with low yielding, but you have the amino acid ready to be used on your medicinal chemistry project. This is the main advantage.
Yes. Thank you.
It makes a difference between having or not having it.
Yeah. And so you talked a bit about changing different molecules around also in terms of green chemistry. How important is that, and how is that influencing decision-making in the industry as a whole?
Now,
So Paolo has frozen on me. Can you still hear him, Jose? Okay, so maybe let's just wait to see if Paolo comes back. In the meantime, I'll maybe address the same question to you, José, as you meet with so many of our clients. How important do you see green chemistry being in the work that researchers are doing?
Paolo is back.
There you are, Paolo. Yes.
Sorry, but
Well, that's quite all right. We lost you at the start, though, so that's maybe you can talk a bit about the importance of green chemistry once more.
Now, green chemistry is becoming an impact factor of decision, because starting from the scratch, if you still having a look on medicinal chemistry project, you also facilitate the future work of process chemistry, where probably green chemistry is more important. But introducing new reactivities, you also facilitate this work for process chemists.
Yeah. Another question. The work you presented just now, it's in close collaboration with academia, tapping into your experience, being the main benefits, but maybe also limitations of working closely together with academia, particularly in translating innovative chemistry into more practical applications.
Okay, this is a challenging question. Okay. The benefit is that in academic world are born probably the most impactant new ideas. The problem is how to integrate them into an industrial environment, okay? Because when you are introducing a new idea, it is quite skeptical, the environment, but also because industries is very strictly to respect of timelines, while sometimes in academic world is not so used to have a strictly deadlines. Okay, why in industry? Yes, this is probably the most challenging factor in collaboration, and also the IP properties and legal.
Which I guess is where from a you can bring knowledge to the table there as well. Yeah, excellent. Thank you so much again, Paolo and José, for your wonderful presentations. Thank you for the audience for asking questions and being here with us today. There is a short 5 in the chat. It's the blue box, and you'll help us your feedback. Session will end shortly. Just stick around, wait, and the platform will automatically go into the next session. Thank you so much for being here. Have a wonderful day.
Thank you.
Bye.
Bye.